So this video is going to go into basic inheritance and Punnett squares. So just a little bit of recap from the textbook. And that's just to remind us that everybody has genotypes and phenotypes. So remember, the genotype of an individual is the actual alleles that they have on their chromosomes. And that can be a big A, so homozygous dominant, big A, which would be the heterozygous, or little a, which would be our homozygous recessive. Now, these two, the big A and big A, they're going to have the same phenotype because the big A allele is going to show. So let's say this is for achondroplasia, achondroplastic dwarfism. So because both of these people have the big A and achondroplasia is dominant, anybody with two bigs or one big, one little is going to be dominant in terms of their phenotype. So their phenotype would be dominant, right, which would mean then their phenotype is that they would be a dwarf. Whereas our individuals that are little a, these individuals are going to have the recessive phenotype, which means that they're going to have a normal height. So when we look at this in terms of inheritance, an individual who is big A, when they make their sperm or their ova, all they're going to be able to give to their kids is one big A. Okay? They're going to give their kid one big A or the other big A, so this one or this one, but they're never going to pass down both of them. Okay? This is something that we call Mendel's law or principle of segregation. Okay, so Mendel's principle of segregation, meaning that in an individual's genotype, they're going to pass down one allele or the other allele to their offspring, but never both of them. Okay, so Mendel's law of segregation, one or the other, not both. So with our individual who might be big A, they're going to pass down either a big allele or a little allele, but not both. So then when we start to do Punnett squares, okay, we have our little square here. And let's say we're crossing one individual that's heterozygous. So there's that, let's say that's mom. Okay? And we're going to cross mom with somebody who is homozygous dominant. So big A, okay, is going to be dad. So we put the alleles on the top and on the side. So we'll put mom on the top, big A, and dad on the side, big A. Then we simply drop down and pull across. So big A for that square. Come down again and across again. So again, big A. Going across to our next box, big A, and our last box, big A. So this is just for a standard dominant recessive trait. The big A being the dominant and the little a being recessive. So those are the easy ones. Now let's look at something that's a little bit more complicated, which is what we call co-dominant traits. So with co-dominant traits, instead of having one dominant and one recessive, we actually have two dominants. And we can see this either as a co-dominant or incomplete dominant. And basically what we're looking at here is that the heterozygote is actually a third phenotype. So in this, we might have big A, we might have big A, big B, and we might have big B, big B. And this is something that we see with our blood types. Type A blood, type AB blood, type B blood. And this is going to be a co-dominant trait. So this individual makes the A proteins, this individual makes the B proteins, but this person makes both A and B proteins. Okay, so that's a co-dominant. They both show up at the same time. Now the Punnett square, however, is going to work exactly the same way as our previous one did. So here we have our Punnett square. Okay, and let's cross somebody who is homozygous dominant A, so big A, with someone who is heterozygous, that AB. Okay, and we fill in the chart the exact same way. So we pull across and we pull down, we pull across and we pull down. So we end up with big A, big A, big A, big A, big A, big B, big A, big B. The biggest thing to remember with codominance or incomplete dominance is that the letters are always uppercase because they are all dominant alleles. Okay, Now for our second type that's a little bit off is what we call our sex-linked traits. Now with sex-linked traits, these traits are on the X chromosome. So sometimes you'll hear of them as X-linked traits. And our notations then are a little bit different. So one sex-linked trait that we could be looking at is colorblindness. So for this, we'd use XC as our allele that's colorblind. A normal X would just be a normal X with no superscript, and then a Y for males being the Y chromosome. So the options then for an individual being colorblind would be XX, two normal X's, XCX, that individual is not colorblind because this sex-linked allele here is recessive. Okay, so sex-linked traits are recessive. Okay, so since we have one normal, it's going to mask our recessive. For a female then to be colorblind, she needs to be XCXC. For a male though, because they only have one X, they're either XY, normal X, normal Y, or XCY, in which case this individual is going to be colorblind because there's no additional X to block this affected X. So for a female to be colorblind, she needs 2 affected. For a male, he only needs 1. So when we do a Punnett square for a sex-linked trait, we have to use the X's and the Y's. So we're going to put our male on the side, we're going to put our female on the top. Okay? And so our traits are always pretty much going to look like this in terms of our Punnett squares. The question is going to be where our superscript letters are. So let's say we have a female carrier, this individual here. She has one allele but not the other, so she is not colorblind, with a normal male. So that means we're going to have a normal X, XC, a normal X, an XCY, and a normal XY. So of these children, this kid, normal, no problem. She's normal, no problem. She is also normal with no problem, although she is a carrier, she is not colorblind. But this child right here, he's going to be colorblind because all he needs is 1 affected X. This is what we call being hemizygous for a trait, as opposed to homozygous, 2 of the same, or heterozygous, 2 different. With a male, for sex-linked traits, they're hemizygous. So only traits that are on the X chromosome do we see a hemizygous situation. So at this point, we've looked at 3 different types of Punnett squares. We've looked at just a normal dominant recessive, we've looked at sex-linked, and we've looked at codominant or incomplete dominant. From here, the only other thing that you're going to be looking at in this chapter is going to be blood groups, and that'll be in the next video.